The earliest scientific attempts to sort the natural world relied entirely on what the naked eye could observe. It was a rudimentary sorting algorithm built on macro-level traits. This approach culminated in the Linnaean system, which neatly divided all living organisms into two massive absolute categories: plants and animals. This binary framework relied heavily on superficial features. If an organism possessed a cell wall, it was automatically categorized as a plant. By enforcing this single visual rule, the system forced single-celled prokaryotic bacteria into the exact same category as complex eukaryotic algae. It also grouped photosynthetic plants, autotrophs that manufacture their own food, alongside heterotrophic fungi that absorb nutrients from decaying matter. A taxonomy built entirely on macroscopic observation ignores the underlying mechanics of life. Biological engineering occurs at the cellular level, and a simple binary sort could not process the deep biochemical differences between organisms. By 1969, the classification crisis required a new architectural model. Robert Whittaker proposed abandoning the binary approach entirely. This matrix visualizes Whittaker's solution, a five-kingdom grid comprising Monera, Protista, fungi, plantae, and Animalia. This system processed organisms through multiple strict biological variables. The first parameter evaluated was cell structure, specifically whether an organism is a prokaryote lacking a nuclear membrane or a eukaryote. The second parameter assessed body organization, cleanly dividing single-celled unicellular organisms from complex multicellular life forms. Next, the matrix sorted for modes of nutrition, how an entity extracts energy, and its mode of reproduction. Finally, Whittaker applied phylogenetic relationships, mapping the evolutionary ancestry to establish how these groups were historically linked. By calculating these five parameters simultaneously, the Whittaker matrix successfully established the algorithmic boundaries for all cellular life on Earth. Applying these rigorous parameters clarified the biological landscape immediately. The newly created kingdom Monera was designated as the exclusive domain for all prokaryotic organisms. Looking closely at the schematic, the defining trait of Monera is its non-cellulosic cell wall. Instead of plant fibers, bacteria build their walls using peptidoglycan, a dense structural mesh composed of polysaccharides and amino acids. Moving down the matrix, Protista acts as the taxonomic catch-all for unicellular eukaryotes. This row cleanly isolates single-celled organisms with true nuclei, separating them entirely from multicellular life. The parameters also demanded the extraction of fungi from the plant kingdom. This division was driven by their mode of nutrition. Fungi are exclusively heterotrophic saprophytes, absorbing nutrients from external decay rather than producing their own. Zooming into the biochemical level of this fungal wall reveals a structural difference. It is built from chitin, a polymer of N-acetylglucosamine. This is a completely different chemical architecture than plant cellulose. The five-kingdom matrix organizes cellular life perfectly, but it contains a massive systemic blind spot. The parameters demand the presence of a cell, rendering the matrix entirely useless when confronted by infectious entities that lack cellular machinery. Viruses break the parameters. Outside of a host cell, they exist merely as inert crystalline particles. They are obligate intracellular parasites. A virus possesses absolutely zero cellular machinery. It has no cytoplasm, no metabolism, no division. Structurally, it is pure nucleoprotein. This 3D schematic illustrates their outer defense architecture, the capsid. This protective protein coat is assembled from smaller, highly organized geometric units called capsomeres. The capsid's only function is to protect the internal payload, a concentrated core of nucleic acid. There is an absolute genetic rule that dictates this payload. A single viral genome is mutually exclusive. It will contain either DNA or RNA, but never both. Plant-infecting viruses, like the tobacco mosaic virus shown here, typically house single-stranded RNA. Bacteriophages, the viruses optimized to infect bacteria, deploy a different payload, housing double-stranded DNA tightly packed within their complex polyhedral heads. Because viruses cannot execute any biological functions on their own, they outsource the definition of life. They hijack a host's cellular machinery, using stolen energy to replicate their own code. If a virus operates with minimal machinery, it poses a severe test of biological boundaries. Can an infectious agent be reduced even further? In 1971, plant pathologist T.O. Diner discovered the answer, isolating the next step down in biological minimalism, the viroid. A viroid strips away the viral defense architecture. It has no protective protein capsid. It is simply a free-floating circular sequence of low molecular weight RNA. Yet this naked genetic code alone is highly infectious, capable of hijacking cellular systems to cause widespread agricultural devastation, such as potato spindle tuber disease. But even the viroid is not the absolute floor of biological infection. That belongs to the prion, an agent that entirely lacks nucleic acid, neither DNA nor RNA. A prion is purely a misfolded protein. Its mechanism is simple and physical. Upon contact with a normal, healthy host protein, the prion forces that target to identically misfold. Lacking any genetic sequence, prions trigger cascading neurological collapse. They are the infectious agents behind fatal, untreatable conditions, like bovine spongiform encephalopathy, mad cow disease in cattle, and its equivalent in humans. The capacity to replicate, infect, and destroy does not require a fully formed cell. It does not even require DNA. It can be executed by isolated, fragmented biochemical components. Whitaker's five-kingdom system mapped a rigorous phylogenetic architecture for all cellular life. It defined exactly how distinct biochemical machines are organized. But the acellular edge cases, viruses, viroids, and prions, continue to exist entirely outside this matrix, operating beyond the strict definitions of a living cell. Complexity also exists within the matrix itself. Lichens function by fusing two separate kingdoms into a single operational unit. A lichen operates as a single dual-engine organism. The algal component, the phycobiont, executes photosynthesis to provide a food supply. Simultaneously, the fungal component, the mycobiont, absorbs mineral nutrients and builds the structural shelter. Because they function through an exposed, codependent nutrient exchange, lichens are intensely sensitive to their surroundings. The presence of sulfur dioxide pollution causes rapid die-offs, making them highly accurate environmental indicators. From the rigid peptidoglycan cell walls of Monera to the dual-kingdom synergy of lichens, biological structures consistently bypass static categories. Biological taxonomy serves as a scientific algorithm, constantly refined to map the structural boundaries of the biological machine.